thanks for that introduction. I'm presenting here the work which is VaxCelerate, which centers around our collaborative, what we see as a collaborative venture with Pfenex, so now hear it being pronounced, that kind of epitomizes to me a developing juncture between academic scientists and business development, product development in industry. The project I'm going to describe in our work with Pfenex used to occupy the lives of postdoctoral fellows who basically sacrifice years of their life to generate recombinant proteins. And what's fascinating to me is that I've lived through this sort of transitional and transformational time where the postdocs can now receive these novel proteins in tubes from companies and then work reproducibly on those proteins. And I think it's a huge advance in the way that academic science is being executed. The other key learning point for me from academia was I did used to work in labs and worked for PIs previously who demanded that new recombinant proteins were generated, and I now realize how variable we were at generating stocks of those particular proteins. Sometimes it worked, sometimes it didn't work, and it was often an enormously frustrating process. That is all gone. And I think we're in a very right position to accelerate academic industrial collaborations in the way that I'm going to set out in the structure of the VaxCelerate program. So what I'm going to present is the VaxCelerate program as an overview, how we're moving forward with the Lassa fever vaccine candidate, and also how the vaccine platform has been developed, and to give you an idea of the potential for this platform and how we're moving it forward. The objectives, project goals, how the project team functions, the vaccine testing timelines and standards, and the status. And I think it's worth saying, because I've been asked to present to Pfenex before, maybe a year or so ago, and we've moved so far on that we've lost a lot of the introductory slides that sort of set the project up. The key point from all of those introductory slides was that vaccine development is excruciatingly slow, excruciatingly expensive. And given that a recent infectious disease colleague of mine showed a graph of emerging infectious diseases, new infectious diseases, emerging infectious diseases, basically reaching sort of a turning point in terms of its inflection point. We're just not keeping up with the infectious diseases that are emerging or re-emerging, and we don't have vaccines for them. And DARPA, the center at the Department of Defense, which prides itself on fast and nimble approaches to problem solving, felt that a program had to be developed which addressed this need. I mean, the classic example was the H1N1 flu. By the time pretty much everyone had had the virus, the vaccine came online. Obviously to much national fanfare and all the rest of it, but the fact was the peak of the pandemic had already passed by the time the vaccine was aware. I don't know if any of you have individual stories. I certainly do. I had H1N1 flu, and maybe 6-7 months later a vaccine was available for it. I was like, great, you know, my whole family had had it, all my colleagues had had it, we'd all had it. So the response was, can you actually generate and preclinically test vaccines faster and more effectively? And that's the enablement that we're undergoing, is to do that. The platform has to be flexible, it has to be consistent to address different pathogens. DARPA also stipulated, and I do believe they're the visionary in the field. They were laying out the sort of visionary approach to what is now accepted as virtual collaboration, distributed development, where different groups with different best-in-class expertise work together to get to a result. So rather than taking one, you know, titanically large biotech company and saying, you make the vaccine for us, use a more nimble platform of different partners, academia and industry working together, each with their own expertise, getting it done fast. The other thing was that whatever product was developed out of that network or distributed development had to be efficiently translatable into something that could be used for patients. That's an incredible stipulation in a way, because as we work in science, we develop wonderful blue skies ideas. That's often the furthest point. We sometimes, I think, come up with ideas thinking, well, all of that with regards to translation will somehow be worked out subsequently by people who are definitely smarter than us and certainly more experienced than us. And what the vision was that actually you can start thinking about the efficient translation first from the moment of sort of discovery and the light bulb going on your head that you may have created a new platform technology. And what I like about it is that the entrepreneurs that I work with and the tremendous product developers that I have on our advisory board, the first question they ask about any discovery is, okay, you've got something here, how do you propose to deliver this to the patient? Is this a once-a-day drug? Is this a injection? Can you take this orally? And they're asking it before the guy's even got his Nature Medicine paper published. And I do believe that was one of the central things of working, in a sense, forward-looking towards the translation of whatever platform we could develop into the translation to human studies. The project also set, I think at its time, extraordinary timelines for an academic group, which was all of this had to be done from a pathogen genome all the way to a preclinically tested vaccine in 120 days. Most of us would like to be working in academia for 120 years, and most R01s run for 5 years as previously, and you renew them in the previous generation every 5 years. So running a project with government support that basically had a readout in 120 days was novel in its time. I actually think it led to extremely disciplined work and extremely cohesive group of collaborators working together with the single aim of getting things done quickly. We talk in the platform development about the HSP subunit constructs and the HSP. That was the light bulb moment between Jeff Gelfand and myself and Tim Brauns and others who are listed on the IP, that a component of the heat shock protein could be used as the core component of a vaccine, and this was our first interface with Pfenex. Just quickly to talk to the component of the distributed development. Basically, we created a wheel with interchangeable units involving at least 5 different companies. You see Pfenex there with regards to vaccine component generation, 21st Century Biochem that generates our peptides, University of Washington that did the protein design, Epivax that screened the pathogen genome for immunogenic peptides, and MPI Research where the GLP animal preclinical studies would be done. So interestingly, and I think it's sort of an interesting lesson in itself, DARPA wanted an academic unit to be at the center of this hub, which sort of surprised me. I said, well, don't you want business types at the center of this, like biotech at the center of this? They said, no. Academics are less likely to favor any particular biotech group, biotech company. We hope that they'll be more critical about interchanging performers and non-performers than biotech companies would. I don't know whether that's true or not, but there's been a lot of pressure from DARPA to say, who are your performers, who are your non-performers? If you have non-performers in your distributed development, switch them out and let's get someone in who can perform. It's a very nimble and somewhat aggressive model, and it has worked to have academia in the center of that, picking and choosing. The other components, the IP agreements, CDAs, MTAs, had to be put in place to operate this. Quality control, GLP, GMP, internet-based communication technologies, and project management. The thing I would say with regards to our relationship with Pfenex is they got all of this. They were a party within that got all of these components and got, number one, how quickly it had to be done, how critically it had to be done, how we had benchmarks of success that we had to report to DARPA, and if we weren't on time, we would get dinged for it. And this was coming from a position where DARPA had a least favorable view of Mass General as an organization that could work fast. So we reversed that, and in 3 years, we have actually changed that position completely. They view MGH and Vic as an operator that can move quickly and nimbly. I've lost it.
Tap the slide.
Oh, I see. Tap on the slide. Got it. Okay. Roles in the project, Epivax, as I just briefly mentioned, was identifying the immunogenic peptides, Pfenex expressing and purifying the self-assembling protein, which I'll come to what the structure of that was under early phase GMP. I think there's a highlight there to be made that on this path to ready translation, in the first phases of the project, we were to do things under near GLP or early phase GMP to show that if we did actually prove principle, we could actually move it into full GMP and GLP for the actual practice of the platform. And I think that was another visionary view that academia could organize that type of thing and could be vigilant about those aspects, and Pfenex was a willing partner in that. 21st century with early phase GMP for the peptides, MPI research for the complete GLP performance of assays associated with vaccine efficacy. The flow of the work basically fits into this, where we get this should really be a pathogen genome delivered to us as a live fire test from the Department of Defense, fed into Epivax, peptides synthesized. In the meantime, the construct protein expression, the Pfenex, bringing them together under GMP at Vic to produce the vaccine itself, self-assemble it, and then do the in vivo efficacy assessments at MPI with data analysis and reporting, all within the 0 to 16-week timeline. The goals were to ultimately provide this confidence and efficacy endpoints and include data potentially for a subsequent IND. The raw materials being produced at Pfenex and 21st century under early phase GMP, which was accomplished, vaccine assembly at Vic under GMP, and then, as I said, the MPI work for the transgenic mouse study under GLP. Now I'm just going to talk about all of the more detailed work which relate to the protein and the construct under the specific vaccine project that's ongoing currently at Vic, and that is there's a significant outbreak of Lassa fever in Nigeria that affects a large population, and which the DoD would like to intervene. They have a lot of interest in Nigeria, and the problem is that there is no vaccine for Lassa fever. We've been working for decades for this, and so they felt that the vaccine that we were sort of putting together as a platform would be relevant. It's a T-cell vaccine, and they thought that it was a good test system. Now, while that was ongoing, we ran experimental academic studies of our platform with an experimental vaccine being ovalbumin, and then a more relevant pathogen, which was influenza. So we sort of we were looking ahead to running the Lassa study, and meanwhile we tiered in these other experimental systems to sort of probe the system. We looked at vaccine peptide construct design and set selection, and we'll talk here about how the project will move from here in concert with Pfenex. So the Lassa vaccine fits into the same model that I talked before. We had these experimental systems in ova with flu that overlapped with the Lassa and so forth, so that we could generate information that would ultimately assist the key piece of what we were doing, which was a Lassa fever vaccine. This is the protein that was the sort of light bulb moment for the inventors, that MTB, which was Mycobacterium tuberculosis, heat shock protein, could serve as a broadly immune activating adjuvant. It's a complex protein. It's 62 kD. It's got an ATPase binding domain. It's got a substrate binding pocket. It's got a lid region which can close down on a peptide that can actually sit within the substrate binding pocket. And this was the protein that is known to interact with chemokine receptors, CD40. It's sort of like a Swiss Army knife of an immune activating both Th1 and Th2 pathways in its own right. The academic question to the side of it, for those who are interested, is what is MTB doing with a protein sitting in its cytoplasm that does all of this type of stuff? That to me, as an academic, is one of the most intriguing academic points that comes out of it. People write about it, it's published. I don't think anyone's realized that this is part of a pathogen that's coexisted with humans for like millions of years, and it has an immune adjuvant sitting in it. It sounds completely counterintuitive, other than if you follow the wave of literature that shows a sort of spy versus spy war, counter war that occurs between the activation state of the immune system and the way the pathogen is actually tweaking it for its own benefit. We decided to harness the protein for our own benefit, in this case, to adjuvant the effect of a vaccine. This is the simple structure here of the simplified model, Mycobacterium tuberculosis heat shock protein, with a linker fusion with avidin. I'm sorry about the layout here, sort of squished it. But the point is that what we felt with this platform is that you could self-assemble onto avidin any targeting protein that you wanted. In this case, in our vaccine platform, it was going to be biotin-related proteins. Now, though this is an inelegant picture because of the typeface here, what I would say is, if you take that and you say to an academic postdoc, well, can you make this for me? You know, most postdocs would look at that and go, well, it would take me probably a year to get to sort of test quantities, and then am I going to be able to reproduce it? Well, we went through that before we got to know Pfenex for at least 2 years, where we had postdocs assigned to making this particular protein. And the thing that academics come across is every time we generated enough protein, we would use it up in the quality control to such an extent we never had enough to use for a vaccine. So meeting with the Pfenex team was a godsend to us with regards to what we could then do next. And in fact, the self-assembling vaccine that then is ultimately assembled with the Mycobacterium tuberculosis, the avidin binding, the biotin-related immunogenic peptide was what we ultimately wanted, but without the core protein, the core adjuvant, we wouldn't have been able to get to any of this, basically. I'm just going to talk about the ongoing projects, or they're actually getting to completion, the evalbumin and the flu that demonstrated efficacy of our vaccine in the context of animal models. We understood that we had to biotinylate peptides, and we also potentially had to solubilize them with PEG, and so that was the way we were moving. These are just the description of the peptides themselves. This is one of the assays that we use to quality control for the SAV, which is the self-assembled vaccine. That's when you self-assemble the MTB-HSP70 avidin with the biotin-related peptide. We could demonstrate these shifts on the gel, and since we're now using longer concatenated peptides, the shifts are much greater than that. This runs to 95–96% completion when you just simply mix 10:1 ratio of the peptide, biotin-related peptide, with the MTB-HSP70 avidin produced by Pfenex in a tube. The product is extraordinarily stable, and as I said, it goes to completion very rapidly. The second one is with the concatenated peptides, SAV2, but not the longest peptides that we're now using, which are even longer than the ones that we used there. We looked for ATPase functionality. These were the types of tests when we generated academic amounts. We would go through all of the protein just doing this, but we were able to show that there was ATPase activity as the native protein would predict, present in both the non-self-assembled MAV and the self-assembled vaccines where they have the biotin-related peptides attached to them. Interestingly, the kinetics of the ATPase change when you self-assemble it with the biotin-related peptides. So that was something we learned as a result of this. Importantly for us, the amount of LPS in the protein was extremely low. This is incredibly important in the vaccine world, that you have minimal amounts of LPS in your vaccine, or at least within the acceptable levels in mice of 50 units per injection, and we were well below that for the proteins that were produced by Pfenex, and so their cleanup process clearly was effective in reducing the LPS, given that it's produced in Pseudomonas fluorescens, which should be chock full of LPS potentially. The evalbumin peptides listed here, the vaccination protocol is a standard prime boost, and then mouse spleen and lymph node harvest, and then I'm just going to show you just some highlights here, which were that we got with the the column in blue, these responses for the self-assembled vaccine, where we included peptides that had both a cleavage point for furin and also were self-assembled in this construct with the MPV-HSP70 avidin. And you can see that the control protein, which is the MPV-HSP70 avidin without self-assembly, doesn't generate immunological responses with this particular set of peptides. So this was our first proof of principle that the self-assembled vaccine could actually induce antigen-specific responses. And the comparison here that's coming up here is with the OVA with CFA, IFA. That's the standard immunological adjuvant that you mix in to make, with your peptides, and that's sort of your positive control. So we got some responders which were above our positive control. Obviously, like in a lot of mouse experiments, there were non-responders, but we were pleased to see, positive responders here. The takeaways from this is we could see firstly that the self-assembled vaccine self-assembled. Secondly, it retained its functionality, which with regard to the ATPase activity. Pierre Leblanc also did other studies of the protein to make sure it did correctly self-assemble. He used a biotin-related HRP to look at that. And then, if you include a PEG and a furin cleavage sequence, having increased the ability for the peptides to be presented correctly, that's our working hypothesis. And then after vaccination, we could see immune antigen or peptide-specific immune response in the lymph nodes of these animals. Obviously, we needed large numbers to do studies that would be statistically significant. In our influenza project, we use now concatenated peptides as well in SAV2. We use the same prime boost. We use peptides that have been identified previously in a bioinformatics screen by Epivax, so that we had sort of peptides that on their own right with CFA, IFA we knew would act to create an immune response, and then tested them in this particular case in a transgenic mouse that has HLA-DR3, which is sort of a human DR molecule sitting on the surface that allows presentation of those peptides. And here again, with SAV2, we saw responders, low responders in the spleen, high responders in the lymphocytes of the lymph nodes. This is a intradermal vaccination, so we do expect to see lymph node responses predominant over spleen responses. We did also see some responsiveness as a broad immune adjuvant in the context of interferon gamma responses for the non-self-assembled Mycobacteria tuberculosis avidin construct. And these showed us that potentially the MAV in its own right could act as an adjuvant, which was kind of, in this particular model with a human HLA-DR3 is sort of important. When we looked at, when we looked at IL-4, what was interesting, we saw, again, responders in the SAV2 in the spleen. In this case, they were higher responders, less of a response in the MAV, which is under here. And then a very dramatic response in the SAV2, which are the self-assembled vaccine with the concatenated peptides in them. And interestingly, again, the SAV2, which has the concatenated peptides, gives you a better response than the ones that just have the monomeric peptides or the single peptides in them. And here, what was reassuring with IL-4 is we saw lower levels of immune activation that was non-peptide peptide specific in a sense. It was just this activating thing. So this is our first clue that SAV2 could generate a peptide-specific response that was quite distinct from what the adjuvant in its own right could do in this particular system. So the takeaways from the influenza was to take the concatenated peptides, continue with the intradermal vaccination. We've seen predominant lymph node response over spleen. We realized that the MPV-HSP70 avidin had an adjuvanting effect in its own right in the HLA-DR3 mice, which actually if we go back to the previous slide, was reduced in the SAV1. So when you put a peptide on the end of this that doesn't evoke a reaction, you can actually silence the broadly immune activating activity of the MTB HSV70 avidin. So that was a useful thing for us to know for controlling experiments in the future. So we know that the VaxCelerate vaccines have the potential here to induce a balanced vaccine response, both interferon gamma and interleukin-4 in these HLA-DR3 transgenic mice. So our move into the Lassa system was to use furin cleavage sites, which we showed were beneficial, PEG spacers, and longer concatenated peptides instead of single ones. In the control group, we assembled the MTB HSV70 avidin with a irrelevant biotin-related peptide to control for the non-specific immune effect. And we increased the number of mice in the group, and we've done the Lassa immunological testing at GLP at MPI as opposed to in our own lab. Now, our challenge with the MTB HSV70 is loading enough peptide into the protein. At the moment, we're loading maybe 2 or 3 peptides onto one protein. So in order to expand that, we began to think of 2 options. One, which we talked about, which was multi-epitope peptides, and the other one was to use dendrimers. And here, working with Dendrotech and 21st Century, we started to look at this type of model where you'd self-assemble the Mycobacterium tuberculosis HSV70 avidin with a biotin-related dendrimer that had multiple peptides that would cover more of the space in terms of the pathogen genome than the concatenated peptides. That hit a roadblock in terms of just pure reproducibility and the overall ability to generate a standard dendrimer, which the Dendrotech and 21st Century are currently working on. And these were the issues that we faced with dendrimers, and any input here with regarding to companies or other ways to make a more standardized dendrimer would be helpful to us. At the moment, we don't know whether we are able to load all of the peptides onto our current concatenated peptides that we need. Certainly with Lassa we can, because of the limited number of peptides we need to actually load in to give you coverage immunologically for that particular disease. But a dendrimer might help where you need more peptides represented to cover the space of the immunological components of any other particular pathogen. So we think that the dendrimers still have potential, but it's still, for us, it's a work in progress. So the current solution going forward with the Lassa vaccine that Pfenex are very much involved with, they produce the MTB HSV70 avidin protein for this particular project fast and on time and at high purity, and with all the QC done by them and by us that confirmed its activity, has gone into the current tested vaccine. The little bit of academia here is that Epivax screened the Lassa genome that we were sent about 100 days ago for potentially immunogenic peptides for HLA-DR3, and came up with a whole list of them, to both the external GP1, GP2, which are the glycoprotein external coating of the virus, and to the L protein and nuclear protein. Interestingly, the nuclear protein immune responses to the nuclear protein have always been associated with bad outcomes for patients. By the time you get an immune response to the internal workings of Lassa fever virus, it's not good. It actually generates an overzealous immune reaction, which is deleterious to the patient. So we stuck with epitopes associated with GP1 and GP2. The key questions that poses is, could we ever use other proteins that are identified, Lassa L protein, for example, as a vaccine target? We know that NP protein is controversial. We currently give decent coverage for GP1 and GP2 within our current vaccine. And this is the workflow more clearly laid out as it was actually executed between Epivax, generating sequences, and also they have a license to generate these HLA-A2DR3 mice. The MPI doing the GLP study, Pfenex generating the assembling protein, 21st century generating peptides, and then MGH coordinating that work and self-assembling the vaccine under GMP with University of Washington, that I'm going to mention in a bit, because that could also interface with the work we do with Pfenex in a moment, what we call the fast-forward vaccine platform sort of development piece. And the Lassa vaccine, exactly as before, prime boost, we have a negative control, peptides alone, self-assembling protein alone, and then the full vaccine. And in this case, we've capped what we call the MTB-HSP70 avidin with an irrelevant biotinylated protein. These are the development path beyond that. We'll report out on this Lassa vaccine project at the end of September. We're in the middle of it at the moment, but obviously the things that we want to talk about potentially with Pfenex are to do with manufacturing, CMC, stability studies, safety tox studies, initial PD dosing studies, and so forth, to see whether this is a translatable product or not. And that it would be side by side with the regulatory approach to whether, what would be the regulatory path of this particular platform. And then ultimately evaluating the protective efficacy in primates, which are the standard sort of pre-human test animal to look at. I just want to mention this because Pfenex is in your audience. This is where we're going from here. The MTB-HSP70, I put this up before. It's a complicated protein with multiple plates and multiple sites in it. There has, interestingly, a region that is critical for LPS binding that actually binds into that pocket. There's a possible CD91 binding site. There's a T regitope that generates potentially suppressive responses positioned around the molecule. There's an ATPase domain and obviously CCR5 binding site as well. We want to try and strip this protein down to the key parts for the broadly immune activating function, and even potentially take out the immunosuppressive components if there are indeed some. So we'll start to, with the help of University of Washington, David Baker there will model all of our constructs as we start to express them, to show that the actual intrinsic structure of those proteins are going to be correctly folded, and if we make deletions, what we may have to add in to stabilize the structure, and then work from the full protein down to these subdomain structures. And there is, at the moment we're contemplating this being done in collaboration with the protein expression core at Mass General that can work, what they say they can work fast. They can get us these constructs in 6 weeks in a yeast expression system, and we would like to get small quantities of this, of each of these subunits of MTB-HSP70, to just do some simple biological assays on to see what functions have retained. And you can see our in vitro assay panel is quite broad for these proteins. If any of them prove to have the functions that we would hope they would do in order to be useful broadly activating immune adjuvants, we would come back to Pfenex and have them produced at the level that we need to then do further in vitro and further in vivo characterization. And this comes back to this first point. You would never have contemplated this 10 years ago, doing this in this way between academia and industry. Now, I have no hesitation to move from a test run in academia to the next phase up with Pfenex to actually express the proteins in a mouse, and those proteins that appear to be valid in our assay systems. Our next steps, we call it VaxCelerate 3, and we've been given the green light by DARPA to start planning VaxCelerate 3, which is big for us. It'll be, most importantly, it'll be a bigger budget for what we have to do. We'll want to do some of those tests of the actual platform itself, the PK, PD stuff. We want to maybe explore the testing of the dendrimers and so forth to see whether that would be a useful thing to combine in this particular setting. And these are for the platform evolution. The actual task for the next steps would be to generate with Pfenex enough of the protein to do primate efficacy studies, and also potentially test it in a gated way in a non-BSL-4 pathogen model first. So before we go all the way to Lassa, we might go back to flu and test it with larger quantities in larger numbers of animals, and also include aspects of the manufacturing safety PD and immunogenicity studies that would need to establish this platform as something that could move on towards on a regulatory path. And then, as I've repeated before, coming up with the second generation of the self-assembled vaccine as we sort of engineer that protein to our particular needs. Now, I mentioned it at the beginning about distributed development. The thing that I've learned is it really requires an incredibly cohesive and cooperative group of people working together to get things done across distance and often virtually. I mean, I don't think I see Bill more than once a year. So in order to do this, you have to have very, very cooperative and active partnerships. This isn't we just send a letter out or an email out to Bill saying, can we have this protein for 25, for whatever amount, and could you send it to us in a few weeks? We're in regular contact with Pfenex about the protein that arrives, the quality of it, any issues we need, any modifications, and Bill and his team at Pfenex have been extraordinarily responsive to us. And all of the partners in this, including the advisors that we have involved in the project, my own team, Pierre LeBlanc, who leads it, Tim Brawns, who project manages it, Bill Taylor at Pfenex, the Epivax team, the MPI team, 21st Century, Jordan Fishman, the UWatch team, have all been incredibly integrated. And I consider it a reflection of the team's common goal to get a particular project done quickly, cost-effectively, and potentially impactfully for people who are at risk for various different emerging infectious diseases. Last acknowledgement is to our research and ventures and licensing. They negotiated all the terms of all the agreements fast in order to keep DARPA happy, who were funding it. And then we're also supported by philanthropists who, interestingly, fill the gaps of this project. They supported the flu work, they supported the albumin work to make sure that we're in the prime position to execute on a strong project with DARPA. With that, I'm going to leave it for questions. I'm happy to take questions at this point. I may not be able to be on the speaker panel because I have to rush later to a meeting. It depends what time it runs to. But if they're technical or other questions, I'd be happy to take them now. If that's okay, Christy. Thank you very much. Yes.
I noticed that you're using PEG as a spacer between the biotin and the protein. Have you found that the size of the PEG moiety that you use has a significant impact on the efficacy of the vaccine?
So others have looked at pegylated peptides, and that was sort of the consensus length. We haven't had the scope of work to do, 4 versus 6 versus 8 or whatever. That in the literature, 4 it seemed adequate to help with the solubility, and interestingly, in the OVA study, helped with the presentation of the peptide, which is probably related to the solubility as well.
Well, it should have an impact. I mean, PEG itself is known to have an impact on immunogenicity, and I wonder if that is also one of your concerns, because if it's too large, it should actually be introduced.
Right. Yes.
Not a vaccine person, let me say this up front. Did you, I believe you said that heat shock protein is present in humans because the bacteria is just present in humans. Is that accurate?
No, so it's present in the bacteria. It's a bacterial protein present in the cytoplasm of the bacteria, but it's not. There are analogs or homologs of it in the human system. We all have heat shock proteins. So, however, I think the MPP HSP70 is maybe 50% similar to human HSP70, but it's not. It doesn't have identical functions to HSP70 in the human version of the HSP70.
All right. So you think it's enough different that you won't get some immunogenicity sort of response to what's already in people because it looks like something in people already.
So this is part of the safety screen would be to look for autoimmune or other responses. Now, the interesting thing about human heat shock proteins is that antibodies have been detected to your own human heat shock proteins, but they don't appear to affect their function. It's rather intriguing. It's an interesting, rather poorly described part of the immune system. So we can generate immune responses to heat shock proteins of our own, but it doesn't seem to impact their function or our health. There are no conditions associated with autoimmunity to your own heat shock proteins.
And in the mice you were testing, is the ethology less, more?
It's, well, because Mycobacterium tuberculosis is really a human pathogen, there are obviously tuberculosis type pathogens that mice get. It probably is just as much homology between MTB HSP70 and mouse HSP70 as it is to human HSP70.
You saw some non-specific increase in immune response, and that's just to the adjuvant itself, if you will?
Right, so that speaks to the ability of the adjuvant to evoke broad, non-specific responses. What we do find interesting, we're sort of modeling the protein with David Baker, is when you cap that protein, it's a big protein, but you cap it with a bartonelated peptide, that broadly immune activating activity goes, but you still retain the peptide-specific responses. So we know there must be folding changes because we know that the kinetics of the ATPase, which is a fundamental component of the HSP70, changes just by the nature of the self-assembly, which this is additional feedback to the biology and science that as we're rushing through, the product development, other members of the team are working on to understand, follow it. But I agree with them. Many interesting questions that come out of this. Yes, Alex.
Mark, I have a question. When you do this, our big dendrimers,
Yeah.
are you afraid about just generating a particle that's so large and huge that it will just crash out, will be enormously
Yes,
big?
absolutely. I mean, I have, when I first saw that molecule, just the design, I was sort of worried about it because of that. It can self-assemble and actually could form much larger aggregates. Interestingly, what's been absolutely true, and this is important for us as an indicator of the fidelity of the MTB HSP70 avidin, when we cap that with a bartonelated peptide, it only self-assembles at that structure. It doesn't start to tetramerize. I know that from our first studies with the dendrimer, we started to see bigger bands present where the thing had, as you're suggesting, sort of started to self-assemble bigger, because you could attach potentially more MTB HSP70 to that structure, or for whatever reason. And I'm concerned about that. I'm also concerned about the standardization of it as a product. How many dendrimers, how many branches, and so forth, and how many peptides are loaded onto it, and so forth. Yes?
So the use of the MTB HSP70 as an adjuvant, is it known particularly to give rise to T-cell responses as opposed to B-cell responses? So, and if that is the case, was it already known in the case of this pathogen that antibody responses are not going to be very effective?
Exactly. So we're choosing the pathogens on the basis of that. So antibody responses are not predictive in Lassa fever of a response. The T-cell response is the key to control of the infection.
And a follow-up question probably is to go to Pfenex actually. So does Pfenex have ways of actually stimulating antibody responses in pathogens that actually might work through antibody responses?
Obviously, if I had our head in molecular biology, should be much more skilled. What we do is we actually work in collaboration. So innovators have the insights, the biology, coupled with Pfenex team of helping guide, looking through iterations of which epitopes might be. We can help produce multiple versions of it. We certainly advise on manufacturability issues, because as Mark pointed out, the goal of this program is not just proving you can make something that works in a mouse, but you can come with something that's viable that can be commercialized. So hand in hand is not only looking to dial and design biologic response, but can you also mark down a pathway that proves that this is something that can truly be scaled up and produced in viable quantities that could actually result in commercialization.
Yes.
You mentioned that you were doing some analysis of HSP70 to sort of try to control the subunits that you retain.
Yeah, so just on the slide that showed when we start dissecting out the
Yes.
subunits and expressing them, there's a whole series of assays that we've developed to look, given what we know about the whole protein, what we would expect to see in terms of ATPase binding, CD40 binding, CD91 binding, the CCL5 binding, these sort of things, we can actually test what is retained in the structure of the subunit. So we will run each of these subunits against those assays and then chalk off which ones are, are going to be beneficial to us with regards to broadly immune activating subunit for our purposes. I mean, it's protein engineering. It's done. We're not doing it. To be honest, we're not doing it in the same way that we might have done it, 20 years ago. Because with David Baker and the protein engineers, they look at every structure, every subunit, and ask, is it in the same conformation that it would have been in the whole protein? And if it isn't, what modifications do you have to make to the structure in order to retain its previous conformation? So each one will be modified according to those sort of predictions. Now they are predictions, and this has been another fascinating component of the distributed development. Each scientific group and technological group come to the project with their own specific view of the science, as it were. David Baker, who's a protein engineer, doesn't think about the immunology. So we have to, there's a lot of intercommunication between us, and the same with the protein expression guys and the peptides about that type of level of interaction. It's all very well making the peptide, but is it going to be presented? Is it, is the immunology going to matter here? So your question is probably the most complicated that we're approaching, which is, okay, can we predict a subunit that's broadly immune activating based on MTB-HSP70? I don't know at the moment.
So, but the idea that you'd like to make the adjuvant portion of the vaccine smaller.
Absolutely, yeah.
Okay. So you'd like to eliminate parts that are unnecessary.
Exactly, yeah. Great. Thank you very much.